And we know mast cells are involved in a number of different pathophysiologic processes, mostly from the human literature, involved in inflammation, colitis, increased permeability, also associated with some interactions with the nervous system as well. So what our question was how do these mast cells, what's the role of these mast cells in the early weaned pig model? So, because we're finding increased numbers, but we don't know what that really meant functionally to the gut. So what we did, we designed a study where we treated early weaned pigs with sodium cromolyn, which is a mast cell stabilizer drug. And we treated the animal for about 24 hours with this mast cell stabilizer drug, and then measured intestinal permeability. And what we found was that just by stabilizing mast cells in the gut, this was able to eliminate, significantly reduce the permeability defects of an early weaned animal. And what this demonstrated to us was that these mast cells, they're increased during stress, but now they're playing a very important functional role in the leaky gut part of the pathology. And then just a little furthermore, what we found was, if you remember back, I showed some data on how when we blocked the corticotropin releasing factor receptors in the weaned pig gut, that prevented intestinal permeability. We also found a link between blocking the CRF system with mast cell degranulation. So this is a mast cell, again, in a pig gut, 24 hours post-weaning. It's highly activated, it's lost all its granules, but when you treat the animal with a CRF receptor antagonist, it essentially blocks or stabilizes these mast cells. So there's a relationship between the stress-activated peptides and mast cells in the stress gut, and that's something that we're studying in our lab right now. And we've also gone on to show, also in the pig gut, that how these mast cells can regulate the permeability aspects. And we showed that when mast cells are activated or modulated by stress factors that are released, they release proteases and TNF-alpha, which then cause changes in the intestinal permeability. So this is kind of the underlying pathophysiology of leaky gut and stress. So just to summarize here, we found that weaning has long-term changes in intestinal barrier function, and that mast cells are playing an important role in this. If we block them, they block the intestinal permeability, and it's linked to this stress response system, the CRF system. And we know that mast cells, when they become activated during a stress response, they can release different factors such as proteases and TNF that cause changes in the barrier of the gut, which really compromise the integrity of the gut barrier and set up an increased susceptibility to disease. I wanted to cover just a little bit about stress. We talked a lot about barrier function, and this is so critical to disease pathogenesis. But we're also interested in what's the impact between this barrier function and other processes such as nutrient absorption, which eventually is going to drive performance and growth in animals, because there is an interrelationship between the two. So if you look at an epithelial cell here, epithelial cells here, one of the major factors that are driving absorption are these transporters here. So this sodium glucose leak transport, this is driving glucose and sodium, and this is what's driving the nutrient transport across the cell. And similar processes also take place with amino acid absorption as well. And what's important here is that this process requires an electrochemical gradient, because it's using the gradient of sodium to drive this sodium into the cell, and now nutrients are carried with it. And this electrochemical gradient an important part of that is how the integrity of the leakiness of the junction. So if you have an impaired tight junction or leaky gut, you're going to have impaired absorptive capabilities as well. And this can have significant impacts or implications for performance and growth. So just as an example, we did some other studies looking at different types of stressors. And this was a study where we did mixing and crowding stress. And so this is 10 days of just packing grower finisher pigs in a pen at a reduced space allowance, and showed that as far as permeability wise, animals exposed to mix and crowding stress also respond with an increased intestinal permeability. We also measured that sodium absorptive or glucose absorptive machinery in the gut as well. And this is also done by using chambers where we can measure electrical activity across the gut when we expose glucose to the gut. And as you can see here, we have a high absorptive capacity in a control animal, but when they undergo this chronic mixing stress, you get this markedly reduced ability to absorb nutrients now. So now you have impaired barrier function as well as an impaired ability to absorb nutrients. We've also shown this to be the case in other stressors as well. This was another study we did with heat stress, and we see very similar changes. Chronic heat stress in animals increases intestinal permeability. They get increased circulating endotoxin as well. And they also lose their ability to absorb nutrients. So it's not just weaning stress at all. Any type of stress, common stressors that animals undergo, the gut is very sensitive to these stressors, and you start getting changes in physiologic function that are going to affect disease susceptibility, stress sensitivity, as well as the ability to absorb nutrients. So it really does have a significant impact on the physiology and function of the gut. So just to summarize this part, kind of as a whole here, there's a lot of production stressors that can have impacts on the gut. We know that early life stressors induce long-term changes in the gut. And these changes can exist all the way into as they're approaching sexual maturity. So they're lifelong changes when the stress occurs early in life. And again, we covered that mast cells are an important part of this process. So now I'd like to go on, and this is the last part of my talk, to really talk about enterotoxigenic E. coli and some of the work that we've done. We've done some work looking at what are some new insights into the pathophysiology of how E. coli cause disease, and also what's some of the interactions between stress as well with this pathogen. So I know this is going to be covered later. I just thought I'd throw a slide here from the U.S. This was courtesy of Kent Schwartz at ISU. Just some of the frequency or etiologic diagnosis of enteritis in pigs, and fairly recent data. But you can see that E. coli in the U.S. has always been a problem. There's always been other diseases such as rotavirus and PED that come up, and TGE. But E. coli is always one of the second leading cause of enteritis cases in pigs in the U.S. This was a study that we did in enterotoxigenic E. coli challenge. This is post-weaning F18 E. coli. And we know that obviously a pretty severe infection can cause some major reductions in performance and growth rate that are associated with the clinical picture as well. We covered a little bit about this at the beginning, so I'm not going to go, just kind of as a reminder of how E. coli causes disease. We know it's hypersecretion, elaborates enterotoxins, and drives this water secretion, as well as impaired sodium absorption, which I'll talk about. And there's also toxins associated with this, the heat-stable, heat-labile toxins. And then there's also, depending with the edema disease for the post-weaning F18 E. coli, as many of you know, the ability of the particular F18 E. coli to elaborate a Shiga-like toxin is going to determine whether or not there's a significant edema disease associated with the diarrhea. And then the histology and the morphology, fairly unremarkable because it's predominantly a secretory diarrheal disease. There's not a lot of morphological changes like in a viral disease. This is some of the pictures from our challenge trial. You can see all this fluid accumulating in the cecum and then all throughout dilated gut loops after a challenge. But then if you look at the histology during the acute stages, the villi look absolutely normal. There's not a lot going on. But after a chronic infection, you will start to see some blunted intestinal villi. So the secretory aspects of E. coli have long been studied and well worked out, but the impacts on the barrier function have not. So does this also change intestinal permeability? And we did this study where we challenged pigs with F18 E. coli. They were about 35 days of age, as post-wean pigs, and then looked at intestinal permeability. And we did find that just after 4 days of a challenge of E. coli, there is a significant compromise in intestinal permeability. So there's increased intestinal permeability associated with this. Another thing that we began to look at was this nutrient absorptive process, because it hasn't been known whether or not, we knew there was a lot of secretion, but what is happening to the absorptive machinery? And so this is 4 days after an E. coli challenge, and this is measuring absorptive capacity on the Using chambers. You see unaffected control pigs, but then pigs that are undergoing this E. coli challenge have this significantly impaired absorptive capacity as well. But then one thing we looked at, a lot of this research and studies have always looked at just immediate post-challenge events. But we also wanted to see were there some lingering changes. So we also looked at gut absorptive function at 29 days post-challenge. And this is when the animals had actually recovered clinically. They didn't have diarrhea anymore, and their growth rate started to pick up again. And we measured absorptive capacity in their gut. And we still found that even after this early challenge and recovery of clinical signs, they can still have this impaired gut absorptive ability that can last for a long time after. So we thought that this was pretty interesting because there's some changes occurring right now when they're going to in this challenge. And just kind of as an aside here, we also looked at barrier function long term, but we found that the barrier function part of it, from what we measured, kind of recovered. But it was just the lingering absorptive capacity of the gut functionally has changed. So I told you about weaning age and how that can also interact with E. coli potentially. And we did a study to look at this where we looked at pigs that were weaned, early weaned, and these were pigs that were weaned about 16 to 18 days of age compared to pigs that were weaned about 23 days of age. This was done on pigs obtained from a commercial production farm. And we just measured how they responded differently to a later life E. coli challenge with F18. And if you look at the fecal scores here after challenge, you can see compared to the controls, even the late wean pigs, obviously when you challenge them with E. coli, they do develop a significant increase in fecal scores, so they had diarrhea. But if you look at the same pigs that were early weaned, they have this marked increase in fecal scores. So a much more severe diarrheal disease in response to E. coli when they were previously early weaned versus late weaned. If you look at gut injury responses, this is intestinal permeability, and I'm realizing these two are switched, so I apologize for that. But this is the early weaned challenge where they have increased intestinal permeability in early weaned pigs, whereas the late weaned pigs did not exhibit this increase in intestinal permeability after four days of infection. So their gut injury was actually quite diminished if you late wean animal, less intestinal injury. This was an interesting part of this study was that we also looked at the immune response to E. coli. And when you look at the late weaned pigs after E. coli, you can see that they had a robust immune response. They had increased neutrophils, increased IL-6, increased IL-8. These are all cytokines that during an infection, they're important for recruiting neutrophils into the tissues. And this is an important process. Yes, it causes pathophysiology, but it is also important for clearing the infection. You need a robust immune response to clear the pathogen from the environment. So I look at this in terms of as a beneficial response. But if you look at the early weaned pigs responding to this infection, they had this markedly suppressed ability to respond to an E. coli challenge. They had their immune cell activation was reduced, and so it was essentially immunosuppressed. And we've also shown this in other studies where responses to LPS and other types of stimuli to the gut, the immune response is actually quite suppressed in an animal that was early life stressed. And this is also just a picture of the gut. You can see here, this is histology. You see the E. coli binding, but then not so much E. coli in the late weaned pigs and increased neutrophils. So this is just to summarize here. This is, we know that E. coli continues to be a major player in disease, in enteric pathogen. We know that it's long been known that classic pathophysiology is mediated through enterotoxin secretion, but we've learned some new insights into this disease and that it can cause barrier function and also immediate and long-term absorptive defects in the gut, and that stressors in early life can determine, or gut challenges early in life can determine how that animal will respond to a later E. coli challenge. So with that, I'd like to end here with this talk. And obviously this work just takes a ton of people. I'd like to acknowledge my lab, previous and past and current members of the lab, our funding. And this is our laboratory here at Michigan State University. So with that, I'd love to answer any questions that you may have.